sessions. Today we have gathered here to share our knowledge with the leading scientists in microbiology and update with the current trends of research in this field. We have with us four renowned scientists who have significant research experience in the areas of inflammatory and infectious diseases. So before starting the session, I'd like to share some housekeeping rules of this e-symposium. Each speaker will be given 25 minutes for their presentation, followed by a 5 minutes of interactive session of question and answer. Participants are requested to ask questions using the chat box, and panelist member will summarize those questions and forward those to the respective speakers for discussion. Okay, now our first speaker of this symposium is Dr. Firdausi Qadri, who is well known to all of us. She is a senior scientist and head of Mucosal Immunology and Vaccinology Unit, ICDDRB. She also leads the Institute for Developing Science and Health Initiative, IDESHI, and an adjunct professor of Bangladesh University of Health Sciences, and visiting professor of University of Washington, St. Louis, and senior associate of Johns Hopkins University. Now, may I request Dr. Firdausi Qadri to deliver her presentation on inflammation in enteric diseases. Madam, please.
Thank you very much, Dr. Shahidul Kabir, and also the rest of the team, the chairperson, Dr. Naz, and everyone else at Stanford University for inviting me. I have a presentation that I would like to, let me try to, without much problems, let me share the screen. So I'm going to talk about, where can I? What's the problem? You can't see. I've kept it open, yeah. So দেখতে পাচ্ছ তোমরা? দেখতে পাচ্ছ না?
ম্যাডাম এখন স্ক্রিনটা শেয়ার হয় নাই।
আমি তো ওপেন করেই রেখেছিলাম শেয়ার করার জন্য। কি তোমাদেরকে পাঠিয়ে দিব?
হ্যাঁ ম্যাডাম, পাঠাতে পারেন।
কিন্তু শেয়ার হচ্ছে না কেন বুঝতে পারছি না। আমি তো এইভাবেই শেয়ার করি। একটু দেখি।
At first click share screen at the bottom panel. You see the share screen.
Share screen to achi.
And then automatically you will see your screen, and then you click that your slide.
Madam, আসতেছে।
আসছে?
আপনার স্ক্রিন শেয়ার জি ম্যাডাম।
আসছে?
জি ম্যাডাম, আসছে।
আমি তো দেখতে পাচ্ছি না।
ম্যাম, আমরা দেখতে পাচ্ছি আপনার ব্রাউজারটা ওপেন আছে।
এটা তো না।
প্রেজেন্টেশন স্লাইডটা ওপেন করতে
একটু সময় লাগে ম্যাডাম, স্লোলি আসে হাতে।
তুমি কি দেখতে পাচ্ছ? What can you see?
ম্যাডাম, আমরা এখন আমরা
ইমেল
দেখতে পাচ্ছি।
মেল তো দেখাতে চাচ্ছি।
মেল বক্স। ম্যাম, আপনি
বন্ধ করে দিই।
প্রেজেন্টেশন জি ম্যাডাম। প্রেজেন্টেশন ফাইলটা ওপেন করতে আমরা আপনার স্ক্রিনটা শেয়ার হয়েছে। আপনার স্ক্রিনে কম্পিউটার যা দেখাচ্ছে ওইটা আমরা দেখতে পাচ্ছি।
মিনিমাইজ করে ম্যাডাম এই ফাইলটা ওপেন করতে হবে।
ফাইল তো খোলাই রেখেছি।
জি ম্যাম, ফাইলটা ক্লিক করলে হবে। ফাইলটা ক্লিক করে ফুল স্ক্রিন করে দিলে হবে।
বললাম তো।
জি ম্যাডাম, আমরা এখন দেখতে পাচ্ছি আপনার ফাইলটা।
এই যে এটা কিন্তু খোলা রেখেছি।
ওকে ম্যাডাম, নিচে ওই যে পাওয়ার পয়েন্ট একটা ফাইল দেখা যাচ্ছে ম্যাডাম, ওইখানে ক্লিক করেন তাহলে বড় হয়ে যাবে জিনিসটা।
হ্যাঁ, তো করলাম এতক্ষণ।
হ্যাঁ ম্যাডাম, ডাবল ক্লিক
করলাম তো। এটা কি ওপেন ফাইল লোকেশন লিখবো?
ম্যাডাম ওই যে ব্যাক বাটনটা দিলেই হবে, ওই ব্যাক ওখানে যে অ্যারোটা দেখাচ্ছে উপরে ব্যাক বাটন, ওইটাই দিলে হবে। হ্যাঁ
ব্যাক বাটনটা ক্লিক করুন। জি ম্যাডাম, ইনফোতে চলে গিয়েছে, ব্যাক বাটনটা ক্লিক করলে আপনার প্রেজেন্টেশন ফাইলটা চলে আসবে।
আচ্ছা, গুড। এখন হয়েছে?
উপরে arrow
এখন দেখতে পাচ্ছো?
Yes.
হ্যাঁ,
জি
দেখতে
ম্যাডাম,
পাচ্ছো?
হ্যাঁ ম্যাডাম দেখতে পাচ্ছি।
Okay, thank you so much. I'm sorry for creating this problem. Okay. So it's my pleasure to talk to you about তোমরা যদি শুনতে না পারো বলবা আমাকে। It's a pleasure for me to talk to you about something that's very close to my heart. I'm an immunologist and work on basically a biochemist, immunologist, and I work on vaccines all my life. And what I've noticed is that inflammation is extremely important. And it can be harmful in many cases, but it can also be helpful. If you don't have inflammation, you don't get an immune response. So my topic will be on different pathogens that we talk about all the time. Since the major pathogens that we see in Bangladesh until SARS-CoV-2 came in were enteric diseases like cholera and enterotoxigenic E. coli, Helicobacter pylori, typhoid fever causing organisms, salmonella, and all the parasitic infections, hookworm, ascaris, trichuris, and the lot. And we then came to SARS-CoV-2. So there's viral, bacterial, and also different kinds of viral infections. So what happens that we have infections and we have co-infections. It's very seldom in a setting like Bangladesh that you see only one infection. You usually go on and see more than one infection. So if you take a patient with cholera, you will also get enterotoxin E. coli. That person will also have ascaris. So it's a very complicated mechanism. And today I will tell you how we try to diagnose pathogens in our laboratory. So we have used a very sensitive method, which is called the Tachman-Eric card, which we use for looking for 18 or 20 pathogens at the same time. Not that we don't do routine microbiology. And with that, we've seen that rotavirus is very common in children, and enterotoxigenic E. coli is also very common. Adenovirus, Vibrio cholerae, Shigella, and enteroinvasive E. coli. Enteroinvasive E. coli is not common in Bangladesh. My talk today I've chosen two of the topics that I've worked a lot in my life, and that's Vibrio cholerae and enterotoxigenic E. coli. And I will show you things that we've done to understand these two pathogens and the disease they cause. So first, cholera. Cholera is an important cause of severe dehydrating diarrhea, which leads to death if not managed properly. So a person can die within 3 to 4 hours if the person is not rehydrated. People don't die in Bangladesh anymore due to cholera. Almost do not die because we have intravenous fluid and we have lots of ORS use in Bangladesh. Globally, there are 300,000 severe cases and over 1.2 million infections. Both adults and children are susceptible to cholera, which requires hospital treatment. And that is how we work at the ICDDRB, the place where I work. We have a surveillance system in which we know that every year we have 30% patients who come to ICDDRB for treatment. Of them, 30% are cholera patients. And of these patients, 30% are children and 70% are adults. So it's a disease that goes over the whole spectrum, not only at children, but also adults. And adults can be very severely dehydrated. So what does it do? The pathogen is Vibrio cholerae. You are all microbiology students, most of you here. You know that the major pathogen for cholera is Vibrio cholerae O1. It causes severe diarrhea, leading to dehydration and shock within a few hours. This is the pathogen we're talking about. It's a comma-shaped bacteria with a long tail, flagella, and it has 3 major antigens: cholera toxin, lipopolysaccharide, and toxin co-regulated filers. But we have known now that there can be many other pathogens that are involved in the pathogenesis. Cholera toxin results in fluid secretion. Massive fluid replacement will be needed. sometimes when patients come to our hospital, there are IV fluids given by 4 or 5 or 6 channels to immediately rehydrate the patient. Although it's a simple disease, it can kill you. And healthy people can be dead in a few hours. So it's a disease that is non-invasive. It does not enter into the bloodstream. So it's called a non-invasive secretory disease restricted to the gastrointestinal tract. And there's a complete season for cholera. It will now appear, and then in February, March, it will increase, and then it will go down, and then it will increase again. During winter months, there's very little cholera. So this is what we've seen all our lives. See, this is one patient who's being rehydrated with many IV fluid bags. So my life, I've tried to understand host and microbes and to answer many questions. And the questions that we've asked always is, how do we understand enteric diseases better? Have you been able to mimic the virulence attributes of pathogens? I mean, a pathogen that we swab on plates, on mechanical agar plates or in gelatin agar plates from stools, are they the same that was creating a disease in the human body? No, it isn't the same. Do bacteria produce factors during in vivo growth, during the growth inside the gut, in the human intestine, or in blood in the systemic circulation? We now know that the bacteria goes through a massive change when you put it on plates. But when it is in the body, invading you, then it is different. So that is what we need to know. Can DNA microarray techniques and RNA microarray techniques and high throughput next generation sequencing give us better insight? It has given us better insight. So we have always wanted to know what are the factors needed for inducing an appropriate immune response after vaccination, because that is what is very important for us. From COVID-19 vaccine programs, you all have all become very familiar with what it takes to make a vaccine and how that vaccine will be as close to natural infection as without causing any pathogenesis. So we have studied many things in cholera, but also, like I said, I'm going to talk about 2 pathogens. We have studied the adaptive immune response. What is the adaptive immune response? It is the immune response that is triggered when antibodies and T and B cells are activated. And we've tried to understand this. We've looked at natural disuse, that is a cholera patient or an ETEC diarrhea patient, stimulates strong antibody responses, acute and memory, bactericidal antibodies, that antibodies that can kill bacteria, as well as toxin and cholera antigen specific responses become elevated in systemic and mucosal secretions. Although cholera and ETEC diarrhea are pathogens that remain on the gut, but they trigger antibody responses in the blood. And we have shown how it is done. T-cell responses are upregulated with CD4, CD19, and Tregs involved. Then we've done traditional tritiated thymidine of flow cytometric acids with CFSC incorporation using Pasha assays, and we've looked at naive and memory cells. We have heard a lot about COVID-19 and how long the antibody responses will last if they have any memory. And so similarly, we've done that for cholera and these other diseases. We've looked at cytokines, IFN gamma, IL-13, IL-17, upregulated ELISA, cytokine B-DARE, ICS, luminate, all these things have been done. And so, we've looked at the T-cell responses by looking at flow cytometric assay, cytokine ELISA, cytokine bead ELISA, intercellular cytokine staining, B-cell responses, antigen-specific antibodies, and so forth. As you can see, we always look at the blood, but we also look into the gut. And for us, the easiest thing to look into the gut for antibody responses is to look at fecal extract, which is very simple to collect from cholera patients or from vaccines. And we also look at what capacity the body has to make antibody secreting cells in the blood, which is actually a correlate of the mucosal responses. And so that's what we've used a lot. Can I go to the next slide? Yeah. So what we've done is we've looked at in cholera infection, there is upregulation of Th1, Th2, Th17 responses. There's a IF and gamma increases, IL-13 increases, IL-17 increases in blood, and IL-17 also increases in the gut, in the mucosal secretions. There's a upregulation of Th1 cytokine and Th1 CD4 T cells, Th1 cells, and also Th2 CD4 T cells. There's a robust pro-inflammatory cellular immune response. So we see over here, as you can see in the picture, there's a negative controls over here, and these are healthy controls, day 2, day 30, and day 180 after disease. And you can see that at day 2, there are IL-17 expression increases in the lamina propria in the gut, and by very soon, by day 30, it decreases again. That is why in cholera, you don't get an inflammatory response that goes towards a negative response. It's a pro-inflammatory response that helps you perk up your immune response, but it does not give you a pathogenic immune response. And then we have looked at the innate immune responses in cholera. We've looked at whole genome microarray techniques to look at duodenal biopsies taken from cholera patients. We do endoscopy and take away the duodenal biopsies. And as you can see, there is a lot of increase in many transcripts in the duodenum. Of the 21,000 transcripts that were seen by these whole genome microarray, 29 genes were upregulated and 33 genes were downregulated. And of these genes, L-plunk, an innate factor, and all these other lactoferrin and all these other factors that you can see over here were upregulated. So lo and behold, even with whole genome microarray, we show that cholera is not a simple disease. It is a disease that has an important component of the inflammation, which is very important. And so there are genes involved in innate defense. And we've got one antibody, one factor, which is L-plunk1, which is we are studied to determine its role in cholera, which is an innate immune response factor. And we've also looked at antibacterial peptides, cathelicidin LL37, which is expressed in the gut in cholera patients, as you can see over here. And we've also looked at in L-plunk in cholera. We have studied individuals in cholera patients from 76 pedigrees that were enrolled in this study. And we've looked at SNPs in the candidate gene of L-plunk, and we've done transmission disaggregation testing on SNPs using this HaploView version 4.1. And we've done found significant association of cholera with the marker in the promoter region. L-plunk is known to downregulate immune responses. What is the biological significance? To understand the biological significance, our group has again studied this, and we've shown that there's an LPS-mediated TLR pathway is stimulated, and then there's L-plunk inhibits LPS-mediated TL4 signaling. So L-plunk1 is not just a standby factor, but it modulates immune response to Vibrio cholera to regulate the immune response so that the pathogenic factors are modulated. So we know now from our studies that L-plunk is highly upregulated transcript in duodenal tissue. A variant in the L-plunk promoter region is associated with susceptibility to severe cholera in the Bangladeshi population. It is a Paneth cell-expressed immunomodulatory protein that decreases TL4-mediated innate immune response to Vibrio cholerae. The maintenance of homeostasis between pro and anti-inflammatory effects in the intestinal environment is crucial. And it is possible that L-plunk serves to reduce intestinal inflammation to enteric pathogens, especially cholera. And we've also done transcriptional profiling of Vibrio cholera of clinical samples using in vivo analysis of Vibrio cholera and compared them with in vitro growth. As I said in the very beginning, what happens when a bacteria is in the body creating the disease, and when you find it in a stool or in the blood and you culture it, there are many changes. So we've compared stool and vomit, and we've seen that the TCP is a toxin co-regulated pilus antigen, CTB. In the beginning, vomit is the early phase of the infection. Stool is the late phase. And we see that in vomit, there are some antigens that are expressed. And in stool, these components, genes are switched off. So forth, and we've also compared to what is seen in the rabbit model, in the rabbit Indian loop, and very similar things are happening. So from this, we know that in vitro cultured bacteria or even virus will not be the same as that what is happening when it is in the body creating pathogenesis. We've also looked at the intestinal microbiome in the susceptibility to cholera. We have shown that bacteria associated with protection or household or susceptibility to Vibrio cholera in household contacts too, that if a person is sick in the hospital, we go to their home and look at other people in the family and see what antibody responses they have, how are they susceptible to the disease. And from that, we have identified bacteria-associated factors. And we also wanted to see, are these bacteria present at the site of infection during cholera? What are they doing over there? And from that, we've seen that there are changes in the bacterial population after Vibrio cholerae succession. At the infection stage, there's a lot of Vibrio cholerae. At the early stage, you can see that there are Enterococcus, E. coli, Streptococcus. At this stage, there is Bacteroides. At late stage, there is Pivotella, Rhizoburia. And in swab that's taken from the patients, you get all these other pathogens. It's very interesting that Vibrio cholerae alone does not create the disease, but all the other microbiome components that are there in the gut also have a role to play. So if you look at microbiome and cholera and the next steps that they're doing, reduce microbial species is seen in the duodenum after antibiotics, ORS, diarrhea. It may be too late for detecting relevant relationships. We have seen samples. We need to have samples that are more closely approximate the duodenal active infection for sequencing, and we are doing this. So we are trying to collect the microbiome, the duodenal active infection by doing endoscopy and taking the biopsies and looking at these pathogens, and we are in the process of doing this now. So the key points that we have from here is that Vibrio cholera infection in humans provoke an immune response, innate immune response. It is not a simple disease where only antibodies are detected. When I first started my work 25 years ago, people told me don't work on cholera because it is not a very difficult disease. It causes disease, there's antibody response, and then you get protected, but it is not true. I'm glad I pursued my path as I did. So it involves IL-37s and beta defensins, disease, increase of HB2, mRNA in the gut at acute stage when bacteria are attacking and causing disease. There's also increased level of expression of other factors at acute stage of disease, showing involvement in the disease process. So the pro-inflammatory factors are very important. The microbiome also seems to play a role in the protection or susceptibility to cholera. Now I come to the second pathogen that I'm going to talk about. So when you see patients with a lot of diarrhea, don't ever think that it is only due to Vibrio cholerae. Half of it can be due to another pathogen which is very dissimilar to Vibrio. It is an E. coli. You're a microbiologist, you're studying, you know that E. coli is not the same as Vibrio cholerae. They are both gram-negative, but they have many different properties. I have worked with ETEC because it also causes diarrhea, acute watery diarrhea, similar to what Vibrio cholerae causes. It is the most common bacterial cause of diarrhea in young children living in endemic countries like us. Most common cause of traveler's diarrhea. People who come to visit us, from 15 to 35% are sick as soon as they come to countries like us due to ETEC. So it's a global health problem. Annually, ETEC causes 1 billion cases of diarrhea and around 500,000 deaths. And so 280 to 400 million cases in children under 5. And so we've seen that ETEC is very common, most common in small children, but also in adults. Oh, where did I go? And so it is also gram-negative rod-shaped bacteria, O groups. It can have many O groups. Vibrio cholerae has different O groups, but only Vibrio cholerae O1 causes cholera and Vibrio cholerae O139, which is extinct now. But ETEC is very difficult because it has over 100 O groups and 34 H groups. It has two major toxins, enterotoxin. One is LT, another is ST, and greater than 25 colonization factors. Most prevalent colonization factors are CFA1, CS3, CS5, CS6. So it's a very complicated pathogen, and these colonization factors are very important for the bacteria to bind to the intestine. Why can I not bring it down? Okay, so this pathogen also causes very good immune responses. It produces antibody secreting cells, which is a surrogate marker of the mucosal immune response in the gut. It produces plasma IgA responses to heat labile toxin, colonization factor in ETEC patients. It also produces different colonization factors CFA1, CS6, LT pathogens. So then it produces a very good immune response. It also produces IgG responses, not only IgA responses. Like cholera, ETEC patients also have a very good memory B cell responses. And so very soon after infection, say 30 days, ETEC patients mount colonization factor specific IgA responses and toxin specific IgA responses. And so memory B cells are developed by day 30 to colonization factors and also to the enterotoxin. And also we've looked at gut homing antibody secreting cells. This is a very important factor to study, the gut homing responses, because when something is triggered in the gut, then the cells from the gut, the lymphocytes from the gut move to the blood through the lymphoid tissues, and you can track them in the blood. That is why we can study the homing receptors and the gut markers in the blood, which is much easier to collect than collecting duodenal biopsies. So we've seen increased colonization factor and CTP specific responses at day 7 compared to day 2 and day 30 after the onset of illness. So this is our marker. When we can't do duodenal biopsies, we do antibody secreting cells. We've also looked at T cell responses in ETEC patients and to toxins. We've used the double mutant heat labile toxin antigen, and we've seen an increase of CD3, CD4, CD5 RO, the memory T cells at early response. At early convalescence, then acute and late convalescence, that is at day 2, you don't see anything. At day 7, you can see over here, these responses increases. You can see it to LTB, heat labile toxin, and to DMLT. And so this is the things that have helped us understand what to do for vaccines. And we have used these parameters to look at vaccine responses to new vaccine responses of ETEC, which we have done a lot of work in phase 1, phase 2 trials, and are planning to go into phase 3 trials also. So also we have done RNA-seq and enteroid analysis from samples that we had in our collection. And we've shown that data from RNA-seq, many genes are modulated by LT, but among them, highly upregulated genes are CCAMs. This is Dr. Alaullah Sheikh, who's now in Washington University, who has published this very interesting data. These data are reproducible, and these findings in the enteroid system and our CRISPR-Cas CCAM knockout cells have a significant impact of an adhesion by ETEC. Therefore, we would now like to use, this is our new study that we have started, small intestinal biopsies for making sections that we have available from earlier studies on ETEC and validate this with in vitro studies. Our plan is to use the slides to look for ETEC and CCAM expression by confocal microscopy in the future. We've also shown that not only colonization factors, but type 1 pili in ETEC also promote ETEC adhesion to intestinal epithelium. Until recently, type 1 pili was supposed to be of no importance to ETEC, but we have shown with Dr. Alaullah Sheikh and Dr. Jim Fleckenstein from Washington University that they're very important components. And they're required for optimal virulence of ETEC. So you can see that if you don't have type 1 pili, you do not get fluid excretion, you do not get fluid accumulation in rabbit ileal loops, but it is important also for us to have type 1 pili, which was not found to be important until recently. So conclusions are high throughput techniques have accelerated our understanding of 2 major infectious diseases, environmental and genetic factors impact on the pathogen and host and shape the outcome of the disease. Even your O blood group has a major factor to show your presence, your capacity to have disease or not have it. Novel genes and antigens identified need to be included in diagnostic and vaccine development attempts to find solutions to the problems facing us. I just want to end by saying something about SARS-CoV-2 and COVID-19 disease. We did not sit still. As soon as the COVID-19 epidemic started in 2020, our lab switched from cholera and ETEC to a SARS-CoV-2 lab. And we have continued like that, although we haven't given up cholera and COVID and ETEC and all these other pathogens and typhoid that we study. We showed about the first to publish in Asia antibodies against SARS-CoV-2 and the follow-up of Bangladeshi patients. Dr. Taufikur Rahman Bhuiya and from our group played a major role in this publication, and Dr. Tahmina Shirin from IDCR. We showed that patients in Bangladesh, people in normal healthy controls in Bangladesh had no response to SARS-CoV-2 antigen, the receptor binding domain of the spike protein, but symptomatic patients had higher magnitude of responses. If you had COVID-19 positive, but had symptoms, then you had high magnitude of antibodies. 100% mild symptomatic patients showed seropositive responses for IgG, whereas only 45% of the asymptomatic patients, you know that you can be COVID positive but still be asymptomatic. There's IgG isotype of antibodies lasted longer in comparison to IgM, IG antibodies. So IgG is very important. In our analysis going forward, we are analyzing how long these antibody responses last. Our results show that IgG antibody remains elevated over 2 months of exposure. Longer term follow-up is needed. And we have actually, Dr. Taufik has completed 6 months of follow-up or 9 months of follow-up, and we are going to, finish writing that paper also. Okay. So we are now doing a big study in looking at mild, moderate, severe, asymptomatic, healthy controls in Bangladesh. Dr. Fahima and Dr. Taufik are taking the lead on this. We're looking at the systemic and mucosal responses. We're looking at the neutralizing antibodies in cases and controls. We're looking at clinical parameters in blood, like CRP, ALT, AST, ferritin, and all the whole total picture. We're looking at the different pro-inflammatory cytokines and Th1, Th2, Th17, and chemokines. We are doing CyTOF in collaboration with Stanford University to look at a whole plethora of cytokines and chemokines. We're looking at transcriptomics by RNA-seq. We're looking at TCR sequencing. We're also looking at cross-reactive and memory T-cell response by activation of induced markers and expression of OX40 plus CD25. So we hope we have done a lot of things right now. We are sending samples. We're also setting up our own lab for all these techniques, and I'm quite sure we will have very important results very soon, which will be special for Bangladesh. And with that, I would like to thank Dr. Taufik, who helps me in everything, who is our senior scientist at ICTDRB in the mucosal immunology vaccinology laboratory. And I would also like to thank all our partners, the government, NIH, PARC, and IDCR, who has always supported us and with whom we work, and the Mass General Hospital, Harvard Medical School, and you can see all these people who support us in our study. And I'm thankful to Professor Shohidul Kabir. I was not in the country and he kept on corresponding and he was relentless, and he and Dr. Taufikur Rahman could help me to come and present to you today. And I thank the Department of Microbiology and University of Stanford for inviting me for this lecture. Thank you very much.
Okay, thank you, madam, for your enlightening presentation. Now, floor is open for question and answer session. So, the participants, if you have any specific question, I see Dr. Hassan Zaki. You have one question. Can you please ask a question?
Yes, thank you for a very nice presentation. So I was wondering, like, so you showed that L. Planck was highly induced during cholera. So do you see similar higher expression during ETEC infection?
We're actually working on this now, but we saw it a lot in cholera. Although the two diseases are very similar, there are also differences. I mean, cholera is causes a very potent, strong response, but ETEC response, it has two toxins and many different factors. So the responses between the two are not always profoundly similar, but they're different because most of the antigens over there are different. Only the major component that makes them similar is the ETEC, heat labile toxin, and the cholera toxin of Vibrio cholerae. Both are actually have 70 to 80% homology, but the rest of the antigens are very different. So we are looking at all this now.
Thank you.
Is there any more questions left? Anyone? If not, I have one question, Madam. During COVID-19 infection, people have some kind of diarrheal symptom. So what kind of changes actually happen that causes severe diarrhea and people cannot recover actually?
Yeah, so very good question. We also, I showed you the last slide of what we are planning to do. We are actually, every COVID patient, we are collecting stools from them to see what the pathogen is. We're also going to do Tachman-Eric on these stool samples. But the most important thing is that we are actually, SARS-CoV-2 actually does not let any one organ of the body free. If you look at all the publications that are coming out, it attacks every organ of the body. So there is diarrhea associated with COVID in many patients. We know about it. There is a symptom of gut involvement. And our study that we are now doing, the longitudinal study, in a very organized way, we have in all kinds of patients, mild, moderate, severe, asymptomatic, and healthy controls from Bangladesh. So this will be, at least we will have 25 patients from each group, and we are following them up till 1 year. So we will have a lot of information from them. And we are actually culturing the stool, not only for SARS-CoV-2, you know that SARS-CoV-2 can be found in stool also, but also for other pathogens. So maybe we will have a lot of information, but what are the pathogens in the stool when the patient has diarrhea?
Madam, another question is like how long this antibody titer stays for Vibrio cholerae infection, like vaccination and so on. This is one question. I'll ask the next one later.
So this question is very interesting. We studied patients. The first paper that we published, we've looked at patients up till 1 year, but people are reporting that you can detect the antibodies up till 6 months. So Dr. Taufik is now following up most of the patients that we got first time, at least many patients who have agreed to give us blood. So we are following them up so that to look at how long it lasts. But you know one thing, that antibodies do not last forever in your body. You have the memory cells that will last. Next time when you have, the IgG levels will also go down. So what will remain is your memory B cells. And these memory cells will be triggered when you're exposed to SARS-CoV-2 again. So that is our hope, that, that if natural infection does this, we hope, we don't know whether it's doing it. So we are also studying the memory cells. But what we need to know is, will vaccines last as long? Vaccine responses last as long? That is our main worry now.
Okay, do you have any more questions left? Anyone? One of the students asked that why patients they die very quickly? Is it due to the toxin or due to dehydration or any other reason? If
How
they
do
have
patients die?
response after cholera infection.
Why they die?
Yes, the students asked the question.
Dehydration, simple dehydration.
Okay.
Cholera toxin just causes dehydration. And that is the cause of the death, not the toxin, not LPS, not PCP. It's because your body is 3/4 water, fluid. And if you lose the fluid, you can lose fluid in half. When patients come to a hospital, they come with half their body weight lost. That means if a 60 kg person can be 30 kg when he comes to the hospital.
Right.
which is severe disease, and then you have to give him all that. As if even if he has a pulse left at the ICDDRB hospital, you can save him.
Thank you very
That
much.
was a good question, actually.
Yes, this is from the student side. So I think there's no more question at the moment. Thank you very much. And we are happy to have you with us and hope to have you again in some other occasions. So thank you, madam.
Thank you so much. So can I leave now?
Yes, madam. Thank you. Yes, thank you
so
for
much.
your valuable time.
Thank you so much.
All right. So our second speaker is Dr. Hassan Zaki. He earned his BSc honors and MS from Department of Microbiology, University of Dhaka. For his PhD, he moved to Kumamoto University, Japan, where he studied the role of nitric oxide in the host defense against Salmonella infection under supervision of Professor Takaki Akaiki. After completion of PhD in 2007, he continued research in the same laboratory with JSPS postdoctoral fellowship. In 2008, he joined St. Jude Children's Research Hospital, Memphis, in Tennessee, USA, where he worked in Dr. Thirumala Devi's laboratory as a postdoctoral research associate. During this postdoctoral training, he studied the role of cytosolic pathogen sensors in host immune response, inflammation, and immunity. Dr. Hassan Zaki joined the University of Texas Southwestern Medical Center, Dallas, Texas, as a tenure-track assistant professor in 2013. Research in his laboratory is aimed at understanding the molecular mechanism of inflammatory disease. So I'd like to request Dr. Hassan Zaki to deliver his valuable speech on diet, gut microbiome, and intestinal inflammation. Dr. Zaki.
Thank you, Dr. Shahidul Kabir. And also I'd like to thank Stanford University for inviting me. It's my pleasure to present my research today. And I'm also happy to see Dr. Firdaus Kadri Appa after a long time. I was used to working at ICDDRB 20 years back. So she was our teacher, our mentor, and our role model. Anyway, so can you see my slide?
Yes.
I hope so.
Okay.
Yes, sir.
So yeah, I just little bit changed the title to include more kind of the immune side, the host pathogen interaction, which actually fits with that the concept of today's seminar. And you see, I put a like a picture showing that the host microbes and man, the relation. You can see that our body harbors like 100 trillions of bacteria and microbes, and the number of bacteria is like more than 3 times of our body cell. So can you imagine like how many microbes we are actually having? So it's not like our own cell, but also like it's a world of microbes. So definitely this host and this our microbes interaction is very important for our life. It actually determines everything, our disease, and not only disease, our healthy life also depends on how our body reacts with these microbes, right? So one of the area that my lab studies is the gastrointestinal, the host pathogen interaction in the gastrointestinal tract. So in this cartoon, actually, I simply show how actually in the gut, how homeostasis and inflammation actually maintain. So in the left side, I show that in the healthy gut, actually how it maintains that intestinal homeostasis. So you can see the gut microbiomes are reside in the gut lumen, which is separated from the mucosal tissue like laminar propria and all this immune cell located in the mucosal tissue. And they are physically separated by a epithelial layer. So this intestinal epithelial layer, and also there is a mucus layer on the top of this epithelial cell that actually protects our mucosal tissue from these microbes. But if there is any kind of pathological changes, like for example, like infection, for example, like our Kadri Appa, he showed that the cholera infection or ETEC infection. And similarly, there are many other ways to cause some kind of damage in the gut. And during this damage, this pathogen or these microbes go inside in the laminar propria, where they interact with a lot of immune cells like macrophage, like dendritic cell, neutrophil. And when these innate immune cells are interacted with these microbes, they become activated. And activated immune cell produce a lot of cytokines and chemokines. And these cytokines and chemokines, basically, they are very important for host defense against any infection. And also they play a role in tissue repair of the injured tissue. However, when these cytokines and chemokines are overexpressed, when there is excessive expression of the cytokines and chemokine, then they cause inflammation. And if this inflammation is continue for a long time, then we have some kind of chronic inflammatory condition in the gut and cause some kind of disease called inflammatory bowel disease or inflammatory bowel syndrome. So my laboratory study this type of inflammatory disorders for a while. So what is actually the inflammatory bowel disease? So inflammatory bowel disease is a kind of inflammatory disorder of the gastrointestinal tract. So there are several types of inflammatory bowel disease like ulcerative colitis, Crohn's disease. So symptom of this disease is like a chronic abdominal pain, rectal bleeding, body weight loss. Basically this disease is mostly prevalent in Western countries like Europe and USA, but also the rest of the world also having some problem. And this disease, basically, there is no specific cause. There is no like one specific gene or one specific bacteria responsible for the disease. It's a multifactorial disease. I can see that our genetic disorder or in that our gut microbiome, immune response, and other inflammatory triggers all are important in shaping that or initiating the IBD and also regulating the IBD. So IBD and other disease basically in the gut, to understand how actually this disease is developed, we should understand the how actually our body interact with the microbes, right? So you know that in our immune system, there are several receptors who actually sense the pathogen. And those receptors are called pattern recognition receptors. And these pattern recognition receptors are, there are many types of pattern recognition receptors, but there are three major types are toll-like receptor, Regai-like receptor, and nod-like receptors. You can see that the toll-like receptors are the major type of pattern recognition receptors who are actually located on the cell surface or in the endosome. So there are different type of toll-like receptors, and they are activated by different kind of bacterial pattern molecule like LPS, peptidoglycan, flagella, DNA, RNA. All these different type of bacterial component can activate these toll-like receptors. And when toll-like receptors are activated, there is a activation of signaling cascade leading to the activation of some transcription factor. And this transcription factor basically are responsible for induction of all inflammatory gene like all the cytokines that I mentioned, and also type 1 interferon, which are very important in T cell and also in our immunity. On the other hand, there are some other receptors are located in the cytoplasm or inside the cell. For example, Regai-like receptors, MDA5, and nod-like receptors. So Regai is a sensor for RNA. On the other hand, nod-like receptors are a large group of receptors. They actually sense pathogen in the cytoplasm, and once they are activated, they actually initiate different kind of signaling pathways. For example, activation of NA-kappa B or activation of caspase-1. So for a long time, I have been working on these nod-like receptors. So before I go to my work, I'd like to show another slide to focus on the inflammasome, because this term is very important in my the work that I'll be presenting. So inflammasome is a complex. So this is a complex molecule which is consisted of a nod-like receptor like NLRP1B, NLRP3, and NLRC4. All these are like nod-like receptors. AIM2, another sensor, but not like receptors, but another DNA sensor. And when these sensors are activated by different kind of ligand, they form a complex with ASC is an adapter molecule and caspase-1. So caspase-1 is is an enzyme which has played many role in our immune system. So when this complex is formed, it's called inflammasome. And in the after formation of this inflammasome, caspase-1 becomes activated, and active caspase-1 play many role. Sorry. Okay. So and active caspase-1 either clips gasdermin D, and gasdermin D induce apoptosis, or it can clips to important pro-inflammatory cytokine like pro-IL-1 beta and pro-IL-18. And once these cytokines are clipped, this IL-1 beta and IL-18 are released, and they play a role in inflammation. And inflammasome also can be activated by a non-canonical pathway. So the model I use to understand the role of all these innate immune molecules or host pathogen interaction in that inflammatory bowel disease is called we're using a model called dextran sodium sulfate induced colitis. So basically, I'm an experimental biologist. We do all this research in animal model or cell culture system in the laboratory to understand the basic mechanism of disease development. So in this model, we feed mice with a chemical called dextran sodium sulfate, and once after like 5 days, mice start developing colitis, and you can see that DSS basically cause erosion of the gut epithelial barrier, leading to the invasion of the microbes in the gut, where they activate the immune cell and produce cytokines and other molecules. And the readouts of DS colitis is like weight loss, diarrhea, rectal bleeding, colon length shortening, and pathological changes in the gut. So previously, I published a paper showing that the NLRP3 inflammasome is very important for maintaining gut homeostasis, because we see that mice that lack NLRP3 are highly susceptible to colitis. We see that the NLRP3 knockout mice, they die quickly compared to wild-type mice. They lost more body weight, and they have a shorter colon length compared to wild-type mice when we induced colitis with dextran sodium sulfate. In another study, we also found that NLRP3 is not the only inflammasome sensor. AIM2, which is a DNS sensor, also play a role in activation of inflammasome in the gut, as well as maintaining intestinal homeostasis. For example, we see when you induce colitis in wild type and AIM2 knockout mice, we see that AIM2 knockout mice develop more colitis. As you can see, the body weight is significantly decreased, and there is increased diarrhea, colon length was shorter, and a lot of pathological changes in the gut. And here you can see that, so the question is how actually the inflammasome maintain homeostasis or protect against colitis. So we study several mechanisms. So one of the mechanisms is inflammasome actually promote epithelial cell proliferation. So we measure this proliferation using a staining called BRDU staining. So proliferating cell uptake BRDU, and you can see that this brown color staining is BRDU positive cells. In control mice, these BRDU positive cells are similar, but on the other hand, in DSS feed mice, you see there is a defect in the number of BRDU positive cells in NLRP3 and caspase-1 knockout mice. So it suggests that that in absence of inflammasome like NLRP3 or caspase-1 deficiency cause increased, there is reduced proliferation, and this proliferation is important to maintain intestinal homeostasis, because if there is less proliferation, then there will be more damage, there will be more epithelial barrier permeability. So, okay. Sorry. So, and another mechanism that inflammasome actually cause induction of antimicrobial peptides. You can see, so after colitis induction, we see that all these antimicrobial peptides is induced, like beta defensin, LCN2, REG3 beta, REG3 gamma, S108, S109, in wild-type mice, the level of these antimicropeptides, some of these antimicropeptides was significantly higher compared to caspase-1 knockout mice. And you know that antimicropeptides production in the gut is very important to protect against the infection. So to confirm the role of inflammasome in production of antimicropeptides, we treated the gut organoids with IL-1 beta or IL-18. So these are the downstream product of the inflammasome. You see that when we stimulate intestinal organoids with this cytokine, there is induction of all these different antimicropeptides. That means that inflammasome actually helps production of these antimicropeptides. And because of this, we see a lot of changes in the gut, like gut microbiome, because we see that NRP3 and AIM2 and caspase-1 knockout mice has increased E. coli. So this is, and you know that E. coli is one of the key pathogen in the gut. So this is very interesting because we know that AIM2 and caspase-1 knockout mice are highly susceptible to colitis, and these mice also has higher E. coli. So to confirm that the inflammasome actually play a role in controlling E. coli growth, we treated mice, like AIM2 knockout mice, with either IL-1 beta or IL-18. And then we measure E. coli growth in their gut. And you can see that after treatment with IL-1 beta or IL-18, there is reduced E. coli burden in the gut. And also after 6 days treatment, we induce colitis in wild type AIM2 knockout mice and mice we treated with IL-1 beta or IL-18. And probably you can understand here that AIM2 knockout mice are highly susceptible, but when we treated AIM2 knockout mice with IL-1 beta or IL-18, these mice are resistant to develop severe colitis. That means they are now less susceptible. So that means IL-18 or IL-1 beta treatment actually cause some kind of protection, and most probably by reducing the E. coli burden. So overall, briefly, what I show that this is a summary. You can see that our intestinal epithelial or macrophages has a lot of inflammasome sensor, and when inflammasome is activated, inflammasome produce IL-1 beta and IL-18, and these two cytokines play many beneficial function. For example, they help proliferation. They also produce a lot of antimicropeptides, and these antimicropeptides control some of this microbial growth, for example, like E. coli, what we showed in our study. So that observation that inflammasome deficient mice has increased E. coli is very interesting, because we know E. coli causes diarrhea, and E. coli is also a pathogen responsible for colitis or IBD. So then we are interested to understand how actually inflammasome regulates E. coli growth. And when we study and we understand that E. coli, the major nutrient of E. coli is simple sugar like glucose and lactose and fructose. So this sugar, and E. coli doesn't have any kind of enzyme to digest complex polysaccharide. So this is very interesting. So then we ask whether this inflammasome deficient mice, which has higher E. coli burden, is there any more nutrient source for E. coli or not? So to understand this, we feed mice with glucose, and after 24 hours, we measure glucose level in wild type AIM2 and caspase-1 knockout mice. And you can see that at the basal level, that glucose level also high in the gut of wild AIM2 and caspase-1 knockout mice compared to wild type mice. And after feeding those mice with glucose, you can see that still the glucose level is significantly higher in AIM2 and caspase-1 knockout mice. And this higher glucose level is higher in not only in feces, but also throughout the gastrointestinal tract. Different part of the gastrointestinal, we see higher glucose level in caspase-1 knockout mice. So remember that this mice has now higher E. coli, and probably this higher glucose level in the gut, probably one of the key factor that helps the E. coli grow in their gut. So this key information is very interesting. And then we ask a question: does high sugar play any role in colitis? And this is really very interesting and very important, because these days we are more kind of we like Western diet. We are used to having all this Western diet in our daily life. And the sugar consumption rate is increasing, particularly in Western countries. In all these Western countries, the person, the sugar consumption is higher than any other country. Actually, more than 100 grams of sugar is consumed per person in Western countries. Whereas the World Health Organization recommend that the per day sugar should not be consumed like more than 50 grams. So that means we are actually consuming more sugar in every day in our life. And this is also very relevant to the IBD in terms of the IBD, because if you see the geographical distribution of the IBD prevalence and incidence, you see the most of the Western country like USA, like European countries, there is more incidence of IBD compared to the Asian country like China, India, or African countries. So that means there is some connection of the Western diet with the IBD incidence. So these background information basically help us to take a detailed study to understand what's the role of the sugar in colitis or IBD. So we did again the simple thing. We feed mice with 10% glucose, fructose, or sucrose for 7 days, and then we induce colitis with the dextran sodium sulfate and measure all this colitis phenotype. And here is the result. You can see this is the study design. So we feed either a control group or we feed the mice with the glucose for 7 days, and then we colitis with the DSS plus glucose. And you can see here that when mice were treated with glucose, they develop severe colitis. And within 3 days after DSS treatment, mice become so sick that we have to just stop those mice and sacrifice those mice because they are almost like moribund. And you can see that the diarrhea score was significantly high compared to the control group. And if you look at that, their colon morphology, you can see that this is the control colon architecture and this is glucose-treated mouse colon architecture. You see the difference. So this is completely full of blood and there is no stool, and the colon also become very shortened. And pathologically, you can see the changes in the gut. You see that in the control mice, there is very nice epithelial architecture. But when mice are treated with glucose followed by induction of colitis with the DSS, there is huge damage. All this lamina propria architecture was lost, and there is huge inflammation. And this is not only because of the drinking of high glucose, because high glucose also basically provides you more calorie. So to understand that this high calorie is not the responsible, so we adjust the calorie. So we made like high glucose diet. This is a solid diet, the pellet diet. And we also provide another group with control diet, matching with the calorie. So that means they are getting, both group gets similar calorie, but the calorie source of this control diet is different, not the glucose. Then we after like 7 days 2 weeks of diet administration, and we then induce colitis with 2.5% DSS. And you can see the mice that were fed with high glucose diet developed more colitis. They lost more body weight compared to mice with that fed with control diet. Similarly, we see that there is significantly higher clinical score like diarrhea and rectal bleeding, and there is huge damage in the gut of high glucose diet. If you compare with the architecture of colon architecture of control diet and high glucose diet, you can see the difference. The complete colon epithelial layer is completely lost, and huge inflammation and damage ulceration. So the question is whether this is only happens to glucose, or what about the other sugar like fructose or sucrose? Yes, all three sugars actually are bad. And we can see here in our next experiment, we treated mice with glucose, fructose, or sucrose, and then we induced colitis with DSS. And you see that all these sugar, three different sugar treated mice develop more colitis compared to untreated mice. And if you pathological score and if you see the colon length, everything suggests that all three sugar induce more colitis and develop severe colitis in mice when they are treated with these sugars. And to confirm this result, we actually use a different model. So this is genetic model. So previously we used a chemically induced colitis model. Now we use a different model called IL-10 knockout mice. So IL-10 is an anti-inflammatory cytokine. It actually helps to maintain gut homeostasis. So if you don't have IL-10, then you are susceptible to develop some kind of inflammation in the gut. So the scientists develop a mice called IL-10 knockout mice, which actually develop spontaneously colitis after 4 or 5 months after their birth. So we use this genetic model, and we feed those IL-10 knockout mice with the glucose after 4 months of their birth, and then we monitor their body weight and look at their pathological change in the gut. And you can see that the mice, IL-10 knockout mice, who are fed with glucose for 5 weeks, you can see they develop more colitis. There is more incidence of colitis development in those mice, and they lost more body weight. There is more pathological changes. You can see the crypt length is significantly increased, and there is huge inflammation inside the lamina propria, and also this the mucosal tissue becomes swollen, and there is also this more inflammation here in this area. So overall, these mice, these genetically susceptible mice, become more susceptible to colitis when they feed glucose. So here, actually, we didn't use any kind of chemical to induce colitis. These mice spontaneously develop colitis just by drinking high glucose for a couple of weeks. And the question is, how actually the glucose trigger colitis, right? So this is an important question. So at first we try to understand whether drinking this high glucose cause any kind of pathological changes in the healthy mouse. That means without any colitis induction, is there any change done by glucose? So we feed mice with 10% glucose or fructose, and then we see the pathological changes in the mouse gut after 7 days. And architecturally, we don't see any difference. They are architecturally, they're very nice, they're fine. We measured the apoptosis by tunnel assay, and this is a green cell is apoptotic cell, and you can see there is no increase of apoptotic cell in glucose and fructose treated mice. Similarly, we measured the mucin production by staining the MARC2. So MARC2 is a gene responsible for mucin production, and you see the red color is MARC2 positive cells. So there is a similar level of MARC2 positive cells in control and glucose treated mice. So we also measure activation of some signaling pathways responsible for induction of inflammation, like I-kappa B and ERK. These are transcription factors that are responsible for induction of inflammatory cytokine. So phosphorylation means, the phospho means this activation. So you can see that control and glucose group of mice, so each individual lane correspond each individual mice, and the protein level of phospho-kappa B, phospho-ERK, phospho-P38 are kind of similar. So no change. And similarly, the induction of these different cytokine are very similar in control and glucose fructose treated mice. So this is very interesting that, okay, like 10% glucose or fructose for 7 days does not have any kind of adverse effect in your gut. So then we focused on the gut microbiome. So we actually collect the stool DNA, and then we sequence them for the bacterial cysteinous rRNA. This is conserved sequence for the bacteria. And then we analyze those microbiome sequence profile in the gut microbiota after sugar treatment and untreated mice. And you can see after sugar treatment, the microbiota composition is shifted. You see this is basically the sugar treated mouse microbiota composition, and here is control mouse. So they are distinctly separated. That means there is a shift of the microbiome microbiota composition. And this can be better understood with this diagram. You can see here that we actually compare the microbiome composition of the sugar before and after sugar treatment in the same group of mice. So we took like 8 mice per group in a group, and then we, before sugar treatment, we collected their stool DNA, and after sugar treatment, we collected the stool DNA, analyzed the microbiome composition. And you can see there is a shift of the microbiome. Particularly, it is interesting that this Akkermansia, this color is Akkermansia, is significantly increased after sugar treatment. On the other hand, lactobacillus, this sky blue color, you can see is reduced after sugar treatment. So here is the statistical analysis of different bacterial family, and you probably understand that some of this bacterial group taxonomy, they actually either increased or decreased after sugar treatment. For example, like a Sutrella increased and Akkermansia increased. On the other hand, Lactobacillus is decreased and Rickenelacea decreased. So there is some kind of alteration. And this kind of alteration is not only for glucose, but also we see the similar alteration in fructose and sucrose. So we compare 4 different group of mice like control, glucose, fructose, and sucrose. We see the similar pattern of microbiota composition in glucose, fructose, and sucrose treated mice. You see there is increased Akkermansia. So this is Akkermansia, and reduced lactobacillus, and also you see there is increased bacteroids. This red color is bacteroids, and this bacteroids is very important, very interesting organism, because it is several of these bacteroids like Bacterus fragilis, Bacterus subtilis, vulgatus. All these bacteroids are actually involved in many kind of gut disease like colitis and colorectal cancer. So looking when we try to understand about this, what is the role of Akkermansia and bacteroids, we see a common feature of Akkermansia and Muciniphila, and they are actually mucus degrading bacteria. So what is actually mucus? You know, in our intestinal tract, there is a layer of mucus at the top of the epithelial layer. And this mucus layer actually protects our epithelial layer and the mucosal tissue from the bacteria which are located in the lumen of our gut. And you can see this bacteria don't cross this mucus barrier. So this is inner mucus layer. This inner mucus layer is completely like sterile. And if you look at the control and glucose treated mouse gut by H staining, you can see, so this is a gut intestinal epithelial layer, and this is our fecal content. And this fecal content is separated from this mucosal tissue by a gap, white gap. So this is basically this mucus layer. You can see very nice gap, and they're nicely separated. So that's why the bacteria, they don't cross this mucus barrier and go inside this lamina propria. But when mice are fed with glucose, we see that this gap is diminished, and this and now the fecal content and this microbiomes are now very closely located to that the epithelial cells, right? And here we also do another staining like the MARC2. This is the mucus staining, and you can see this is the mucus layer in control mice, and this mucus layer is very nice and thick. But in the glucose treated mouse gut, this mucus layer become very reduced and very thin. And because of this thin mucus layer, you can see now the bacteria, they can cross this epithelial barrier during colitis. So here actually we stain the bacteria with a red color, and you see in the control mouse, these bacteria are nicely separated from this colonic epithelial cells and lamina propria by this mucus layer. But in the glucose treated mice, this bacteria is now very close to the epithelial barrier, but they didn't cross yet. They just separated by a very thin mucus layer. But when we induce colitis in those mice, you see that is still like after day 1 or day 3 in the control group, this bacteria is still nicely away from this our mucosal tissue. On the other hand, in the glucose treated mice, these bacteria now start invading. If you can see here that the red color, that they are now inside this lamina propria, and after day 3, there's huge infiltration or penetration of this bacteria into the lamina propria and in the mucosa. And when these bacteria go inside, they actually activate the immune system. They activate that our epithelial cell to produce a lot of cytokine and chemokine, which actually cause this colitis and inflammation. So now I would like to little bit focus on the mucus. You know that the mucus is a glycoprotein produced by our cell, and after synthesis of the mucin actually glycosylated in the endoplasmic reticulum and Golgi apparatus, and then secreted. So this is this is structure. So this is a complex glycoprotein. And so there's many bacteria in our gut. They actually can degrade this mucin by producing a lot of enzymes. And this enzyme can degrade those of this bond of that mucin structure, this complex polysaccharide. So there is those bonds can be cleaved by some of this enzyme produced by this bacteria like Akkermansia, Muciniphila, Bacterus fragilis. And they produce all these type of enzymes responsible for degradation of the mucin. And since we observed Akkermansia and Bacteroides, their abundance is higher in glucose-treated mice. So we try to understand, okay, whether this bacteria cause this degradation of this mucin by producing this enzyme or not. So that's what we measure this enzyme that are responsible for mucus degradation, like sulfatase and acetyl glucosamidase and fucosidase. And you can see here that that level of this enzyme was significantly high in glucose-treated mouse feces compared to the control feces. On the other hand, some other enzyme, which is not responsible for mucus degradation, but they are involved in degradation of the plant carbohydrate, like galactosidase, glucosidase, and xylosidase, those enzymes are not defective. Those are kind of similar in both control and glucose-treated mice. So to confirm this role of the gut bacteria in colitis, we actually did a very elegant study. Here, actually, what we did, we feed mice with glucose or left untreated. And after 7 days, we collect a stool from these control mice or these glucose-feed mice and transfer into germ-free mice. So germ-free mice don't have any bacteria. So now we've put this bacteria from either either control group of mouse or this glucose-treated mouse. And then this is recipient mice. So now this recipient mice has bacteria either from the control mice, or this group of mice has bacteria from this glucose-treated mice. Now we induce colitis in all 4 group of mice. This is recipient mice and this is donor mice, right? Now you can see that this one is that glucose fecal recipient mice. So they develop severe colitis compared to that mice that received from control mice, right? So they are pretty resistant compared to this this GFR, that is that recipient mice that received microbiome from that glucose-feed mice. And if you compare that gut their intestinal length, you can see that the glucose GFR group of mice, that means the mice that received a microbiome from glucose-treated mice, they develop more colitis. Their colon lengths are shorter. There is more pathological changes, and they have more body weight loss. So overall, it suggests that the bacteria is the key factor, because when we drink high sugar diet, it actually causes induction or enhance the growth of some of these pathogenic bacteria, and those pathogenic bacteria basically responsible for higher inflammation and colitis development in our gut. So we recently published this paper in Science Translational Medicine. I didn't show all this data, but if you are interested, you can have a look in this paper. And so what is the message? So message is like, we have to be very mindful to our diet, right? So because every day we are taking a lot of different kind of food, but we also, we like all this sweetened food, sweetened beverages, right? So, but the sweet is, definitely we like sweet, but we have to be careful at the amount of sweet we are taking, because they can be harmful to our gut. They can cause that degradation of the mucus layer. And when this mucus layer becomes thin, the gut bacteria go inside in our laminar propria and mucosal tissue, causing activation of the immune cell. And this activation of immune cell cause inflammation and leading to induction of colitis. But this inflammation can be a trigger for many other diseases like obesity, metabolic syndrome, neurodegenerative disease, and cancer. And as you know, like nowadays we have increased cancer, increased obesity and diabetes, all these metabolic disorders. So we really have to be careful about that high sugar diet. I mean, we cannot avoid, but we should limit our diet with high sugar. So these are the people who actually did all these studies, basically mostly the sugar study. Dr. Shahanshah Khan, he was the postdoctoral fellow. Dr. Sumay Waliullah, both of them are equal first author in this paper. And Abdul Wadud Khan, he helped me analyze the microbiome part. And thanks to the funding agency and our collaborator. I'll be happy to take any question if you have.
Thank you, Dr. Hassan Zaki, for your informative and elaborate presentation. So there are a few questions. We are running actually beyond the time schedule.
Yeah, I know. Yeah, I take
so, some of the participants want to know that whether Coca-Cola, if they take it on a regular basis, can it cause cancer? Number one. And number two, can it cause a change in microbiota of the intestine? And how about the metabolites the microbiota doesn't change with the food? These two questions first. Okay.
Yeah, no, definitely. You know that the 250 ml Coca-Cola can contain about like 37 grams sugar, right? So now our World Health Organization who recommend that for a healthy person should take like 25 grams sugar per day. And it should not be more than 50 grams. Maximum you can increase to like 50 grams for a healthy life. But if you take like suppose 2 can of Coca-Cola or any soft drinks, you're already like 80 grams of sugar, right? And we also have many other things like cookies and other all this food. We also get sugar from there. So definitely that if you take like the soda drinks, definitely it will cause kind of alteration of gut microbiota. And in our study, we show, yes, this high sugar diet basically altered the microbiota composition. And if this is like, if you continue, if you have a chronic pattern of taking high sugar, definitely those microbiome will cause a permanent damage in your gut, right? So like colitis. And when there is a consistent colitis or chronic colitis, then you may develop colorectal cancer, because chronic inflammation is a trigger for cancer development. So there are not many studies on this type of research, but recently there are studies coming up that role of high sugar in IBD, colorectal cancer, and other inflammatory disorders. So definitely, I would say that too much taking of all these soft drinks is not good. I mean, we can take, but we should, minimize.
Thank you very much. Another question is if there is metal in the food, items, can it cause a change in the microbiome and similar kind of problem?
I'm not sure about the role of metals, but you know that different metals may have some role in microbiomes in different way. So some of this, in our gut, there are a lot of different type of bacteria, and they have different metabolic and nutritional requirement. So some bacteria, suppose like simple sugar, some bacteria like complex sugar, some bacteria may, their growth may enhance by some kind of particular metal. So it will be, I think, interesting to see if those metals has any effect. But to my knowledge, I don't have any idea like whether there is any effect of these metals on gut microbiome.
Right.
But
another question is the mucus layer. It becomes narrow due to some mucolytic enzymes you had mentioned.
Yes, yes.
But is there any other relationship between sugar intake and narrowing this layer of gut?
Is there any other?
Any of the causes?
Yeah, I mean, so that mucus layer thinness is one of the cause, but definitely there is other mechanism. For example, some of this bacteria.
No, the layer becomes thin due to the mucolytic enzyme you mentioned.
Right.
Is there any other factors that can reduce this layer?
Not to my knowledge, that's not for mucus layer thickness, there is no other cause. So this is the major cause, because some of this bacteria produce mucolytic enzyme. But on the other hand, so we also see there are some pathogenic bacteria, their growth is enhanced, right? So those pathogenic bacteria, they can penetrate our mucus barrier. Even mucus layer is intact, but they can penetrate that mucus barrier and epithelial barrier if those bacteria like ETEC or EHEC or some other pathogenic bacteria, their growth, they have some capacity to penetrate. So I would say those growth of those pathogenic bacteria is not good.
Okay. The last question is one of the participants asked that whether a ketogenic diet or other low carb diet can reduce inflammation and help healthy life, because in previous days when sugar was not used, now maybe before 50 years, our previous generation, they didn't used to have these kind of fast foods and exactly. They had a good lifestyle. So do you have any suggestion that a ketogenic diet or low carb diet can help?
Yeah, definitely, and you rightly say that basically this IBD and all this metabolic syndrome like obesity, all this kind of problem is a contemporary disease. So previously, our old generation, like 100 years before, probably they don't take this kind of high sugar diet or anything, so they don't have this kind of problem. But because of our diet, now we have all this kind of inflammatory and metabolic disorders. And yeah, definitely ketogenic diet may help, but I think we should also need some more research to understand its effect, because if you change anything like natural anything may not have some good effect. I mean, definitely it may provide some good effect, but it may also have some other effect, but we don't know. So we have to be careful. I think the best thing is to maintain a balanced diet, right? So the balance is important. In nature, everything is balanced. So if we take the balanced diet, I think that's the best thing.
Okay, I think we are running short of time. Thank you very much for being with us. And this occasion, we would like to invite you again in some other occasions.
Yeah, sure. Yeah, I'd like to.
Please stay with us. So thank you very much. Now I'd like to request our next speaker, third speaker, is Dr. Chatyana Dendi. Am I right?
Yes, Chaitanya.
I think the speaker is present here. Okay. So he received his PhD from Department of Biochemistry at the Indian Institute of Science, Bangalore. His PhD work focused on understanding the mechanism of action of curcumin in combination with alpha-beta artemether for the treatment of recrudescent and cerebral malaria in mouse model. His current research focuses on the role of vitamin A in gut lymphocyte function, and he is currently working as a postdoctoral fellow at the Department of Immunology in the University of Texas Southwestern Medical Center. Today, he will share with us his research finding, and which is on epithelial retinic acid receptor beta regulates serum amyloid A and vitamin A dependent intestinal immunity. So please.
Can you see my screen and hear me?
Yes, okay.
Yes, sir.
Sound, can you please increase the volume a bit?
Yes. Okay. So so today, so before I talk about my work, I would like to thank the organizers of this e-symposium. So in this presentation, so I'm going to talk about how vitamin A plays a role in intestinal immunity by controlling a epithelial cell-specific protein called serum amyloid A. So first, so before I dive into this into this topic, so I'll tell you so what kind of work we do in our lab. So I would like to thank Dr. Hassan Zaki and Firdausi for basically laying a very good platform for my talk. So you have seen this slide in Dr. Zaki's presentation. So human colon harbors like trillions of bacteria, and they're very important for controlling the human physiology. So in particular, immunity and metabolism. But very little is known about like what are the molecular mechanisms, like how this microbiota is able to control host physiology, immunity, and metabolism. So we don't know. So our goal is to, our lab goal is to basically obtain so fundamental and molecular insights into these processes. So without further ado, so I will jump into this topic. So we know that microbiota. So it is like most like in trillions, it is colonized in the intestine. So it is highly like present in the mainly present in the colon, and it is also present in the like terminal part of the small intestine, and even the little bit in the proximal part of the small intestine. So this microbiota So they have a symbiotic relationship with the host, but yet they're able to cause disease in a susceptible host. So they can cause inflammation, and in severe cases, they can cause sepsis also. So sepsis is a life-threatening inflammatory condition. So, so the important question in the field is, so why don't our intestinal bacteria constantly make us sick? Because we have a lot of bacteria in our system, but they don't cause disease all the time. So why? So that is one important question. So our lab is interested in answering this question. So by focusing on the epithelial cells. So here you are seeing the cartoon of the intestine, and so this intestine. So this is so I'm showing you the micrograph of this intestine here. So so intestine has a single monolayer of epithelial cells. Basically, that entire intestine is covered by these epithelial cells. So, and it faces the microbes, and it protects the immune system from the microbes, so which are present in the lumen. And so we are very interested in these cells because so they're at a critical juncture between the microbiota and human immunity. So we wanted to know like how these cells are able to like so manage these microbes so without causing disease to the host. And indeed we find that these epithelial cells are very important in this process. So so in the next couple of slides, I'm going to tell like how we study these microbes and how we study epithelial cells in our lab. So we have mice so that does not have microbes, which we call germ-free mice, and we also have mice so that have microbes, and which we call conventional mice. So we have techniques to isolate intestinal epithelial cells from either germ-free mice or conventional mice, and we studied we did the DNA microarrays in old days, but now RNA sequencing, and so we find that more than 200 genes are upregulated when microbiota is present. So which is telling us that so basically microbiota is inducing these genes, and so we find that antimicrobial proteins like Rh3 gamma and delm beta so are secreted into the lumen. So as mentioned by Dr. Zaki in the previous talk, so this lumen has a mucus layer. So this mucus layer basically separates the bacteria from invading the host epithelial cells, and so these epithelial cells, if something crosses this this mucus layer, so these epithelial cells make these antimicrobial proteins. So whatever like crosses this mucus layer, so those bacteria are killed by this antimicrobial proteins. So basically, so Rh3 gamma and delm beta are bactericidal protein, and these two proteins like kill gram positive bacteria, and so we also find out another group of proteins called serum amyloid A. So these are also induced by the gut microbiota, but the role of these proteins is not known clearly, and we also know that this serum amyloid A protein expression in the intestine requires dietary vitamin A. So here I am showing you a micrograph of mice small intestine, so that have vitamin A fed diet and vitamin A deficient diet. So we clearly see that when vitamin A is absent from the diet, so this serum amyloid A protein levels, so that is shown in the red, is completely absent when vitamin A is not present in the diet. And when vitamin A is present in the diet, we clearly see this signal in the smallest intestinal epithelial cells, and it is true with in the case of liver also. So the serum amyloid A proteins are produced by intestine and liver, and the serum amyloid A proteins are in the intestine promote production of IL-17 by TH17 cells. So Dr. Dan Littman group is working on this topic since very long time, and they have made similar contributions in this topic. And so what I'm trying to show here is, so serum amyloid A is basically produced at the site of dietary retinol uptake and at the site of retinol storage. So since it is serum amyloid A production is mainly present in these two tissues. So we have focused on vitamin A because these two tissues have very important role, and vitamin A plays very important role in these two tissues. So vitamin A is acquired by the dietary sources, and retinol is the major form in the retinoids, and so this retinol is converted into the bioactive metabolite in the intestinal epithelium, and this bioactive metabolite is transported to the immune cells like CD4 T cells, so which are equipped to secrete different cytokines, and IgA producing B cells. So IgA is very important for maintaining gut immunity. So basically, vitamin A is very crucial, and so retinol, so which is a metabolite of the dietary vitamin A, so is fat soluble, and it must be transported by binding to the protein because it is fat soluble. So it cannot be transported on its own. So it needs some protein to be transported into the tissues. So the important question is how retinol is mobilized to the immune system. So from gut epithelial cells to the immune cells. So how this retinol is transported is not known. So, and also another point to be noted is in normal conditions, so at like steady state, so liver like basically receives this retinol from the intestine by the retinol binding protein. Basically, at the steady state, retinol binding protein transports retinol and circulates retinol to the tissues that require it. But during infection, so this retinol binding protein levels are reduced drastically, and during infection condition, retinol requirement is more, but this retinol binding protein levels are decreased. So what proteins circulate retinol during infection is not known. So so we asked this question, and so we found that serum amyloid A, so which is a microbiota inducible protein, and also it can bind to retinol and basically regulate adaptive immunity. So so we know that retinol is transported to immune cells in the intestine by the SAAs, but how? So SAAs that transport retinol, so how the gene expression of SA is controlled is an open question. So in order to answer this question, so I already shown you this slide. So we got hints from our previous study. So vitamin A, dietary vitamin A basically controls the SAA expression. So we wanted to study like what component of vitamin A is doing this job. So in order to do that, so we used a Modke cell line, so which is a cell line that is derived from mouse epithelial cells, basically replicating the mouse intestinal epithelial cell model basically. So we treated these cells with either retinol alone or LPS. So I have shown you that so microbiota as well as dietary vitamin A is important for the SAA gene expression. So in order to recapitulate that, so I have added retinol in place of vitamin A, and I have added the LPS in place of microbes. So we see that when both of these are present, this SAA gene expression levels are increased very significantly. And when I added BMS 493, so which is an inhibitor of this retinoic acid signaling pathway, so which means though retinol is present, so the signaling cannot happen. So that's why so we see a huge reduction in the SAA expression. So we conclude that SAA expression requires retinol. So in this slide, I'm going to talk a little bit about how retinoic acid receptor signaling occurs in the epithelial cell. So epithelial cells absorb dietary vitamin A, and so retinol is the major form, retinoid form in the vitamin A. So this retinol is converted to retinol and retinoic acid by these enzymes called retinol dehydrogenase, and this retinoic acid is the bioactive metabolite, and this metabolite translocates into the nucleus, and in the nucleus, so this retinoic acid has these receptors called RAR, retinoic acid receptor. And they have three isoforms, alpha, beta, and gamma. And so this retinoic acid receptor heterodimerizes with RXR and induces RAR-mediated gene expression. So when these receptors are bound to retinoic acid, so they bind to a promoter element called retinoic acid responsive element in the promoter of its target genes and induce its gene expression. So this happens. This is how this signaling happens in the epithelial cells. So in our next experiment, so we have tested like what kind of retinoic acid receptors are present, and we found that retinoic acid receptor beta is very dominantly present in the epithelial cells, and alpha and gamma are present in the immune cells like macrophages and DCs. So since retinoic acid receptor beta is present in the epithelial cells, so we wanted to delete this retinoic acid receptor beta in the epithelial cell specific manner. So in order to do that, so we have crossed RAR beta flox mice to Williams-Cree. So when we cross this flox mice to Williams-Cree, so it generates a mouse model. So that is RAR beta is deleted specifically in the intestinal epithelial cells. So we use this mouse model and check for the gene expression of SAA. So and we find that when RAR beta beta receptor is absent from the intestinal epithelial cells, so SAA 1, 2, 3, all the isoforms of SAA are highly downregulated in this genetic background. And so basically, in simple words, so this mouse model is basically in this mouse model, epithelial cells are blind to vitamin A. So which means though vitamin A is present, so these epithelial cells are not able to see the vitamin A, and signaling does not happen. So in this mouse model. So so I have already shown how retinoic acid signaling occurs, and so since we have shown that retinol is important for SAA gene expression, so we directly tested if RAR beta activates SAA transcription by binding to its promoter. So in order to do that, we did a cheap assay and chromatin immunoprecipitation assay, and find that so RAR beta specifically binds to the retinoic acid responsive element at the 224 nucleotide upstream, and and we find this particular element, and retinoic acid receptor beta is basically bound to this this nucleotide sequence. And when we mutated this sequence, and we find that this binding is abolished, so that is shown in this bar diagram. So which clearly indicates that retinoic acid beta activates SAA transcription by binding directly to its promoter. So since we have this molecular insights of how retinoic acid receptor beta is inducing SAA levels, so we wanted to know what is the outcome of bacterial infection in this mouse model. So, and we find that when we infected these mice with the Salmonella typhimurium to these mice, so we find that this bacterial dissemination is very high in this mouse that are deleted for RAR beta in the intestinal epithelial cell. So we find bacterial burden is very high in the ileum and liver and spleen. So these are the organs that are far from the intestine. So which is telling us that so this bacterial dissemination is very high in this mouse model. So due to the mucosal barrier breakage, and next we wanted to know like how this dissemination or how like why we see more disease in the RAR beta deficient mice. So in order to test that, so we know that vitamin A plays a very key role in the Th17 production. So since these epithelial cells are blind to the vitamin A, so we wanted to know what happens to the production of adaptive immune compartment effector function. So we find that Th17 cells produce very less IL-17A in the RAR beta deficient mice, and when we added recombinant SAA to the lamina propria cells of the wild type or RAR beta deficient mice, so we find that so when we added SAA, so we find that it rescues the IL-22 and IL-17 production by the CD4 T cells. So which is telling us that SAA is basically very important for bridging the epithelial cells to the immune cell compartment. And next we wanted to know what happens to the IgA production in this mouse model. So we find that so IgA producing B cells are B cell levels are decreased in this mouse model. So which is telling us that this epithelial cell RAR beta signaling is regulating the development of IgA production by the B cells in the at the mucosal sites. So with this, I would like to conclude with this slide. So in this short presentation, I have shown that dietary retinol is very important, and so it is basically absorbed by the epithelial cells and converted to the retinoic acid, which is a bioactive metabolite. And this retinoic acid binds to the RAR beta and is involved in the gene expression and in turn protein expression of SAA. And by directly binding to the RAR beta, by directly binding to the SAA promoter and inducing SAA gene expression and increasing the intestinal immunity. So by producing the TS17 and IgA containing and IgA production by the B cells. So with that, I would like to thank my mentor Laura Hooper and my lab members and our funding sources. And I would like to thank Dr. Shahibul Kabir for giving me this opportunity, and also Dr. Zaki Hassan for connecting me to the Dr. Kabir. So with this, I would like to conclude, and happy to answer any questions you have.
So Chaitanya, is there any alteration of gut microbiome if there is deficiency in vitamin A?
Very good question, Dr. Zaki. So we did the 16S RNA-seq on these mice, and we find no differences.
Okay, thank you.
Yeah.
Thank you, Dr. Chaitanya Dendi, for your valuable presentation explaining role of vitamin A dependent intestinal immunity. Do anybody have any question?
Maybe our student have some question on our QA.
Yeah. Okay, there is one question from his student panel, and one of the student asked that, can alteration of gut microbiota be the future of liver disease?
It depends because microbiome is like very complex. So it's like not one kind of bacteria is present. So it is a very like lots of like trillions of bacteria are present, and different classes of bacteria are also present. So so basically, I'm not sure if that helps actually in treating liver diseases.
Thank you, Dr. Chaitanya Dendi, for your valuable presentation. Now, I would like to take over the session on behalf of Professor Dr. Shahidul Kabir sir. Maybe there is any problem in internet or current electricity issue. So our last and fourth speakers of this e-symposium is Dr. Mohammad Jubair. He completed his BSc Honors and Masters from the Department of Microbiology, University of Dhaka, and PhD in Biological Science. He has experience in bacterial pathogen genomics, genomic epidemiology, clinical and environmental microbiology, molecular microbiology, and conservation genetics. Now he will present his talk on nanopore-based metagenomic comparison of airway colonizers between cystic fibrosis patients and healthy individuals. Please, Dr. Mohammad Jubair.
Thank you, Dr. Zareen. Can you hear me?
Yes. Yes, everything is okay.
Can you see the screen?
No, sir, not yet.
Can you see the screen now?
Yes, yes,
Yes, sir.
we can see.
Thank you, Dr. Zerin, for inviting me for the talk, and thank you for the chairman of Stanford University for giving me the opportunity to present my work. And I was fascinated to hear all the talks, the last 3 talks. It was absolutely amazing. I would like to talk a little different since my work is involved with respiratory microflora. So I would like to talk about respiratory uh, microbiome in the light of cystic fibrosis disease. So I'm Muhammad Jubair. I'm a postdoctoral scholar at the Biomedical Science Department at University of Central Florida, which is located in Orlando, Florida. My topic would be nanopore-based metagenomics comparison of airway colonizers between the cystic fibrosis patients and healthy individuals. First of all, cystic fibrosis is not familiar to in Bangladesh. It's a more Western-based disease. So I'd like to talk more the background knowledge about cystic fibrosis to give the audience an idea to see to give the audience an idea about cystic fibrosis disease. Cystic fibrosis is an autosomal recessive genetic disorder which involves a mutation in the cystic fibrosis transmembrane reductase regulator protein or CFTR protein. It causes a dysfunctional transport of fluoride ions across cell membranes among different body systems. This disease is life-shortening. It's a genetic disorder that occurs in 1 in every 2,500 newborns yearly in USA. It has been estimated that 30,000 people living with CF in the USA. The symptoms include chronic lung obstruction, cough, shortness of breathing, bacterial infections, infertility in man, salty sweat, weight loss, malabsorption, maldigestion, pancreatic and lung disease. CF is one of the chronic lung diseases that the disease course is significantly influenced by the change in the composition of the lung microbiome. In the absence of the normal lung microbiota, the patient is more susceptible to bacterial infection. If you see in the figure, the healthy and cystic fibrosis patient airway, if you clearly see the mucus formed in the airway and it causes lung obstruction and shortness of breathing. Healthy lung microbiome predominantly consists of proteobacteria, firmicutes, and bacteroidetes at the phylum level. And we see Prevotella, Fusobacteria, and Bellionella at the genus level. When it comes to CF lungs, we find all these bacteria I already mentioned in the healthy lung, and also we find the pathogenic the pathogens, which are Pseudomonas aeruginosa, Staphylococcus aureus, Burkholderia cepacia, Haemophilus influenzae, MRSA, etc. If you see in the figure, it's at the beginning of the patient when they're born. At the beginning, we see the Staph aureus are more predominant, whereas Pseudomonas compared to Pseudomonas aeruginosa. But after the teenage, or when they get adult, Pseudomonas aeruginosa takes over compared to Staph aureus. The treatment, antibiotics are used for the treatment and also chest massage to clear the mucosal buildup. But until recently, available treatment options have been directed towards alleviating CF symptoms. But recently, a new classification of drugs called CFTR modulators it's been used, which provide an opportunity to slow the progression of the disease. Out of 30,000 living people living with CF in the USA, an estimated of half the people qualify for the CFTR modulator treatment. So these CFTR modulators are referred to as, it's more like a personalized medication, since each modulator is correlated with to a specific CF class. I will not go detail about the classes, but I would like to mention the names of these modulators has been used widely. Ivacaftor is the most widely used. Ivacaftor is the most widely used modulator, and also lumacaftor, tezacaftor, and elixacaftor those are also being used. The potentiators hold the gate on the CFTR protein and allow chloride ions to flow through. The characters, it aid in forming the correct tertiary shape of the CFTR protein, so the protein is able to reach the cell membrane and transport chloride. The objective behind designing the next generation modulators is to develop drugs that can be approved for the CF patients that could have not benefited from the previous modulators, including individual with a single F508 del mutation. CFTR modulators are shown to improve patients' overall symptoms, such as decreasing the number of hospital admissions, decreasing the number of exacerbations. It helps to weight gain. It also helps improve lung function. However, little is known about how CFTR modulator impacts the lung microbiome. CFTR modulators, the research has been done on CFTR modulators, the effect of CFTR modulators on lung microbiome. Those data is limited, inconsistent, and conflicting. Most of the studies, they did 16S ribosomal RNA sequencing and short length sequencing technique. And majority of the studies have only focused on one type of modulators, which is ivacaftor. Here is the comparative. Here I showed the 3 study has been done on effect of CFTR modulators. The first one, the Rowe et al. In 2012-2013, they did the study, and those patients had G551D mutation, which was treated with ivacaftor. Those patients were known of Pseudomonas aeruginosa infection. Overall, we saw they found that in the Pseudomonas aeruginosa abundance decreased in 6 months, and overall decrease of combined CF pathogens and increase of bacteria normally found in healthy lungs. There is another study which is very conflicting compared to the first one I mentioned. The Barnard et al. In 2015, they did a study on 3 CF patients with the same mutation, G551D, and that was treated with ivacaftor. Unfortunately, they found no significant changes before or after the treatment. The third study was Hazard et al. In 2016. They did a study on 12 adult patients with G551D mutation, which was treated with ivacaftor. 8 of 12 patients had chronic Pseudomonas aeruginosa infection. But initially, the Pseudomonas aeruginosa abundance decreased and healthy microbiota abundance increased. But they saw after a year the density of Pseudomonas aeruginosa rebounded. So which is all the study, it is very conflicting because no study is giving all the 3 studies are not giving uniform results. So it is unclear whether the observed changes to airway microbiome are due to the antimicrobial properties of the drug or lung function improvement. This gap may impede clinical decision making related to modulator therapy in conjunction with antibacterial management of respiratory colonizers and pathogen. These are the gaps what motivated us to do a study on cystic fibrosis. So we collaborated with Nemours Children's Hospital, which is located in Orlando, Florida. They have a pediatrics CF center. They have 123 patients who all of them are kids, and they have 45 patients on CFTR already, and they're expecting another 55 patients on CFTR by next year. So we designed our project with 10 of the CF patients if we can get. But unfortunately, when the pandemic started, those patients are very vulnerable to, because their lung is very compromised, so they're very vulnerable to SARS-CoV-2. That's why it was very difficult to collect samples from them. So we were able to, last year we were able to collect only 2 samples, and I'm presenting, in this presentation, I will present these 2 samples, 2 CF samples compared to 2 healthy individual samples. So our hypothesis, we hypothesized that microbial diversity is reduced among patients before the initiation of therapy, with the predominance of select opportunistic pathogens in the respiratory flora. We would like to assess the microbial diversity, which is alpha diversity among CF patients, and compare them to healthy controls. We'd also like to determine the prevalent species by determining beta diversity among CF patients and compare them to healthy controls. Our ideal methodology was to collect the CF, to collect the samples before CFTR, 1 year before CFTR treatment, and every 3 months interval, and 1 year after CFTR treatment, every 3 months interval. That was the best scenario. We wanted to have like 8 different samples and ultimately extract DNA from there and do sequence analysis by nanopore, by Oxford nanopore. But ultimately we only got 2 samples from the 1 year before CFTR treatments. That's what I'm going to present, I'm going to show you today. So the workflow involves, there is a host DNA depletion step, which is very important. Most other studies before, they did not do that host DNA depletion. So what happens when our samples, we collected samples of oropharyngeal swabs, because most of all our patients are kids, and it's very hard for them to give us mucus or sputum samples. That's why we went for oropharyngeal swabs. So here, the most of the samples, we see that predominantly there are host DNA would be like 90 to 95%, whereas bacterial DNA are probably 5%. So if we extract DNA from the sample directly, there is a highly likely there is a chance of missing the bacterial species from the analysis. That's why host DNA, we introduced host DNA depletion or human DNA depletion at the beginning, and then we extracted DNA, which is bacterial DNA and probably a little bit of human DNA mixed. And then we analyzed by qPCR or real-time PCR or quantitative PCR to see how much human DNA was depleted, and also how much bacterial DNA was lost. The best scenario is 95%, above 95% human DNA depletion and very minimum loss of bacterial DNA. Then the DNA sample goes to library prep, which is Oxford nanopore design library prep for our experiment, for metagenomics experiment, and then then the sequencing is done in nanopore MinION sequencing. And then ultimately the data is, we analyze the data, we analyze the data and ultimately we find the results of the lung microbiome. Here is more in details, all the steps in details. So this host DNA depletion, we acquired this protocol from Karalampas and his colleagues. They published a protocol for rapid diagnosis of lower respiratory infection using nanopore-based clinical metagenomics, which we now propose to employ here. Saponin, there is a step we use saponin. Saponin helps to promote host cell lysis and to remove the host DNA. There is sodium chloride used. Sodium chloride is used to deliver the osmotic shock and lysing the damaged host cell. There's the enzyme used, HL-SAN. It's a DNase. It's so this all degrades the host DNA. So performing host DNA depletion allows us to focus on the microbial population, which in past studies where human DNA was not removed. And then after the host depletion, we extracted the DNA using QIAGEN DNA-Z blood and tissue kit. And then we quantify the DNA by qubit. Then ultimately we do the qPCR to see how much DNA, how much host DNA is depleted before and after, and also how much bacterial DNA lost before and after. And then ultimately the DNA goes to, we used the library prep kit to prepare the library and then sequencing with MinION. So the question is why nanopore MinION? There are studies before they did using Illumina MiSeq sequencing, but we selected nanopore MinION because it has the advantage of the ability of providing long read data, which is 1 KB to almost like 1,000, 1 megabase pair, which is very long DNA. To give a little bit to make you understand about nanopore, it has biological pores that are inserted into a synthetic polymer membrane, which is immersed in an electrolyte solution that generates an ionic current through the pores. It has a very small pore to allow the nucleotides to pass through the pore and by applying current. And by detecting that changes made in the current, that as nucleotide pass, we determine the type of nucleotide. We optimized a high resolution nanopore-based metagenomic sequencing, which will allow us to identify the bacterial species present in the respirator samples and characterize the microbial community at an unprecedented level of detail. An additional advantage is the read length. In MiSeq, we usually see in Illumina MiSeq, we usually see 100 to 300 base pair short reads, whereas here we got 7 KB to 2.5 KB to even 70 KB, which is really high for our study. So which we can use to identify species level data, as well as presence of genes of interest like antibiotic resistant genes with high confidence. So we have two healthy, we have oropharyngeal swabs from 2 healthy individuals, MJ and TA, and we have oropharyngeal swabs from 2 CF patients, CF1 and CF2. So we saw from our qPCR data, we saw 99% depletion of host DNA with very minimum loss of microbial DNA, which gives us the confidence to move forward with the DNA to sequence the data, to sequence the DNA. The 2 healthy individuals, we got, for MJ, we got 1.87 gigabases of data, and for TA, we got 1.72 gigabases of data. Whereas for the CF patients, we obtained almost 2 gigabases of data, which is really good, very good amount of data to analyze everything in very detail. So this is the pie chart, which we obtained from the analysis of MGRust. MGRust is an open source web application server that suggests automatic phylogenetic and functional analysis of metagenomes. It is also one of the biggest repositories for metagenomic data. And so the raw sequencing data were uploaded in MGRust, which is an open source application. And we got for the healthy individuals, if you see in the pie chart, we Streptococcus, Prevotella, Fusobacterium, Veillonella, those are the most predominant phylum. When we go to Corona pie chart, Corona is a new visualization tool that allows intuitive exploration of relative abundances and confidences within the complex hierarchies of metagenomic classification. Its rich and interactive displays facilitate more informed interpretation of metagenomic analysis. So we see from the Corona pie chart of MJ, we see more species level detection. And those are all the microbes are usually found in common in lung microbiome, in healthy lung microbiome, so which is good. For TA, for the sample of TA, we saw here we are showing the pie chart and Corona pie chart side by side. The results are pretty similar to MJ. We are seeing Prevotella, Streptococcus, Veillonella. Those are more predominant. And we are not seeing any pathogenic strain in both of these, any lung or CF pathogens in both of these healthy patients, which is very good. Here's the bar plot, if you see here, the side by side of MJ and TA, the healthy samples. We see for MJ, Streptococcus, Prevotella, Fusobacterium, Veillonella, those are more dominant. And for TA, Prevotella, Streptococcus, and Veillonella are more dominant. So it's pretty similar, and this two represents a healthy lung microbiome. When it comes to CF1, the first sample from the CF patient, who is a 12 years old male patient with a known Pseudomonas aeruginosa infection. In the pie chart, we see, the dominant ones are actually pretty same, like the healthy lung microbiome. The Prevotella, Streptococcus, and Veillonella are more dominant in the for the cystic fibrosis 1 sample. But when we will go to the Corona pie chart, we see more detailed explanation, more species level detection. Here is the green part. I will go, even here, it's hard to see all the pathogens I mentioned before that are present in cystic fibrosis patients. So in the green part, if we focus more, then we will go more detail. We are seeing the Proteobacteria family. And here we have a gamma Proteobacteria. If we go furthermore in gamma Proteobacteria, here we see Haemophilus influenzae, 6%. And if we go even further, we have Pseudomonas, which was 5%. We go back. Yeah. So there was 5% Pseudomonas present in CF patient, and out of this 5%, 18% were Pseudomonas aeruginosa. In Enterobacteriaceae, we see here we see other strain, other species level detection, and if we go even further, we see Barkholderiaceae, Barkholderiales family. Here we see that Barkholderia cepacia 4%. In the Bacillus family, we see here Staph aureus present 7%. When we go to the second CF samples, CF patients, who is a 2 years old male patient. Here in the pie chart, we're seeing the similar similar bacteria who are predominant, which is Prevotella, Veillonella, and Streptococcus, but the pathogens are in very small amounts. That's why we are not seeing those pathogens in the pie chart. We have to go to the corona chart. When we go to the corona chart, we are seeing the species level detection. We will go further down this green and blue, this part. We're seeing a Proteobacteria family. Here we see that 1% was Barkholderia. And in the Barkholderiales family, we see that there is a Barkholderia cepacia is 8%. When we go to Proteobacteria, we see Haemophilus influenzae 7%. And the Pseudomonas was present 1%. And from this 1%, we found that Pseudomonas aeruginosa was 21% of this 1%. In Bacillus family, we see for the CF2, we see 3% Staph aureus. In Proteobacteria family, we see here 7% of Haemophilus influenzae. So here is the bar plot. In the bar plot, we see the actually it looks pretty same. At the most dominant ones looks at same, but here when we have less diversity compared to healthy samples. I will show in the next figures. So the summary of all the bioinformatic analysis is for the healthy samples and for the CF samples. It looks the most prevalent bacterial genera are pretty same. Those are mostly Streptococcus, Prevotella, Fusobacterium, Veillonella, etc. Whereas in health samples, we're not seeing any CF pathogens or any pathogens. And in case of CF patients, we're seeing for the first CF samples, we are seeing that 5% Pseudomonas. Out of this 5%, 18% was Pseudomonas aeruginosa. And also we saw 6% of Haemophilus influenzae, 4% of Barkholderia cepacia, 7% of Staph aureus. And whereas for the second CF samples, we saw 1% of Pseudomonas. And out of this 1% of Pseudomonas, 21% was Pseudomonas aeruginosa, 7% Haemophilus influenzae, 8% Barkholderia cepacia, and 2% Staph aureus. So this is clearly shows us before CFTR treatment, and those patients are, they have the Pseudomonas aeruginosa culture infection already confirmed. And we're seeing after the sequence analysis data, we're seeing actually all the major CF pathogens are actually present. Pseudomonas, Barkholderia, Staph aureus, they're all present in these two patients. When we calculate alpha diversity, we tested and found a decreasing microbiota diversity with a reduction in lung function to be a significant change. The loss of diversity was accompanied by increase in microbiota dominance. Subsequently, we stratified patients into lung disease categories of increasing. So when we see the alpha diversity looks at the differences within a sample. So this high diversity, which is expected for the healthy samples, we are seeing that high diversity. And when it goes to CF samples, we see that alpha diversity is low compared to healthy samples. This alpha diversity is measured through MGRAST program. It's like an automatic, the program gives us the value of how much alpha diversity in each samples. Then we compared beta diversity. Beta diversity, which measured the overall change between two samples, was calculated using the vegan package in our programming language. This Bray-Curtis dissimilarity, which is BCD, the BCD values can be ranged between 0 and 1. A value of 0 means that the two samples have the same species composition, and 1 means the two samples do not share any species. So when we see the between two healthy samples, we are seeing the BCD values is 0.23. So that means they're very similar. And between the two CF samples, their BCD value is 0.31, which is expected. That between the two groups, they should have inside a group, they should be similar to each other. But look at this table. When we see that comparison between the CF patients and healthy individuals, we're seeing the value is much higher. That means the two samples are very different. And the two group of microbiota are very different from each other. That's why the BCD values is higher. So in conclusion, here we were able to assess the microbiome of 2 CF patients with Oxford nanopore-based sequencing. Longer read lengths allowed us to get species level resolution. We compared the findings in the CF microbiome to healthy individuals. We compared the alpha and beta level diversity between two groups. In summary, we showed that the microbial diversity is reduced among CF patients before the initiation of therapy compared to that of healthy controls. Our future direction is this project lays the foundation for a more comprehensive study to assess the changes of the CF lung microbiome after 1 year of treatment with modulators. We'd like to assess the lung microbiome at least 8 more CF patients to have the total of 10 CF patients, which will give us a good study and also we'd like to publish it. We'd like to obtain samples obtain samples from patients 12 months before and 12 months after the CFTA therapy. The results from the study will have direct implication to current treatment practices. Our results will elucidate more general microbial dynamics in CF airways, as well as have implication for the future use of nanopore-based sequencing technology for rapid pathogen diagnostics. Thank you all. If there is any question, I will take the question now.
Thank you, Dr. Zubair, for your valuable presentation. Now, floor is open for question. We have several questions already put in the box. So, are pathogens in CFTR patients airborne or opportunistic? Where do lung microbes receive their nutrients from? And cystic fibrosis carrier status may pose increase in risk of COVID-19 and so on. So these questions are first, then we can go for the others.
Lung microbiome is commonly the bacteria for a healthy sample, the common lung microbiome, we find it in our system, in our upper respiratory tract, and even in the lower respiratory tract. Airborne, I'm not sure about that. Is it airborne or not?
Are they opportunistic or airborne?
Yeah, some
are they
of
being
them
introduced from the environment, or
yes, yeah,
kind
some
of
of
genetic
them
dip
can be
allows them to?
opportunistic, introduced from the environment, and when it goes inside, and because of this imbalance of transmembrane channels, it the CF cystic fibrosis patients actually develops that, and those microbes started forming biofilm. It's kind of like they started forming, the pathogens start forming like a.
Where do they get the nutrients from? Where do they get their nutrients when the microbiome changes? Do they have any other sources for their nutrients? Or?
My understanding, when they form the mucus, that mucus probably helps them to get the nutrients from. I'm exactly not sure about it.
Right. So another question is it possible to use lung microbiome as a medicinal approach for cystic fibrosis patients? Like changing the microbiome, like when you eat food, you change the microbiome of your gut. Like, is there any approach or possibility?
I haven't seen any study so far that it changes anything. Because antibiotics actually, sometimes it works, but sometimes it not. This is an area all the research have been done. Still, the treatment is not well established. So the probiotic approach, I'm not sure how it will help to treat the CF patients. But as I mentioned, the CF modulators it's already been used, and there more CF modulators are coming to treat those to treat CF patients, which are designed specific on the mutation, what the specific mutation, and it's a personalized medication, which helps to give the CF patients a better life. Actually, we don't know yet the CF patients are completely recovered.
We take a question from Dr. Hassan Zaki, another panel speaker. Please ask your question.
Yes, thank you, Jubair, for a very nice presentation. So I was wondering whether, so you see that in the CF patient, there is altered microbiome in the lung, particularly some of these pathogenic bacteria. So it is because of this altered pathology in the gut, in the lung, because the cystic fibrosis cause a lot of inflammation and maybe mucus secretion, all these things that may help to grow this bacteria. Or due to this bacteria, basically you have this problem like all this respiratory problem.
It's the mutation happened. It's actually biborn, like the recessive genetic disorder. So the complication starts actually because of the imbalance of the transmembrane channels, and then it creates the environment to colonize those pathogenic bacteria, to opportunistic pathogens to colonize there and form this complication. So this is the combination of the change in the change of the environment because of the imbalance of the transmembrane channels, and also the opportunistic pathogens. So those modulators works against the mutation. They try to minimize those mutations, and that helps to improve the lung, the better lung, better breathing, so yes. But still, I mean, we're not sure that it's completely when it's completely, because some of the studies we saw that it's after a year those bacteria actually rebounded. So it's still a lot of work is going on this, and we'll get more clear idea the more.
Another question is, so you present that the 3 studies showed like 3 different results after treatment. So those studies are in the same population or like different population? So do those studies consider that like the race or environment, diet, all these factors?
It can be the sequencing method they used. Like they used the 16S ribosomal RNA sequencing. That is why we wanted to introduce the Oxford nanopore based sequencing, which has long fragments and which gives us more confidence to get like that we are not missing anything. So I don't know exactly what happened when in their studies, probably those ribosomal RNA sequencing is not giving us the clear details after all this before and after treatment, what's happening. Whereas we did only the before treatment samples, but we didn't get yet. We did not get yet the after treatment. We'll see when we have the after treatment samples. Then we compare. Then we'll get actually a better idea.
So do you think that that lung microbiome can be dependent on like our environment, diet, or any other environmental factors can shape the microbiome in this of the cystic fibrosis patient?
Pollution level, pollution level can be. It's been reported before that when there is pollution in the outside, air pollution can expedite the symptoms of CF patients, and also there is what is called PEB1 in 1 second, how much mucus can be secreted. That is also an indication of how much how severe the disease is, how severe the disease for the patient. So it may be the environmental factors may be involved, but we don't have like details research study on those.
Got it. Thank you.
Welcome.
Okay, another question is like, is the COVID-19 patient, are they more prone to such kind of,
yes,
cystic fibrosis patient?
Cystic fibrosis patients, their lungs are very compromised. So, and that was one of the reasons we got only 2 samples, because no one wants to come to the hospital. Everyone was calling to the doctors, and they were getting prescribed over the phone. So without the patients in the hospital, we were not able to get the vaccine. They're considered as high risk SARS-CoV.
Another question from student: in the physical exercise, is it useful or harmful for cystic fibrosis patients?
I don't know that it is, but there it's been. We see most of the CF patients are like they're very thin. They're because of their constant breathing difficulty. They're not healthy. Exercise probably helps. I'm not sure. I haven't seen in any data on that.
All right, Dr. Chaitanya Dandi, you want to ask some questions, I guess? Do you mind mute and ask?
Hey, Hey, Mahmoud, very nice talk. So similar to what the previous question, so our CF patients are more susceptible to like other like viral infections, especially like rhinovirus or influenza virus infection, and are they more susceptible to like asthma kind of attacks?
So far, the reports, all the studies have been reported. We are not seeing is there any correlation of other viral diseases with CF patients. The CF already is a combination of so many bacteria, so many bacterial pathogens are already in the lung microbiome, in their lungs. So SARS-CoV-2 is, since the compromised lung people are very high risk. So we know that for SARS-CoV-2, in this pandemic, those patients are considered as high risk group, and they try to avoid any exposure to other people. I don't know about the other, you said that common cold or influenza, how they're correlated with CF patient, if they can be opportunistic for CF to cause disease in CF cases. CF, I'm not sure about that. I'm sorry.
Okay.
So is there any more question from the audience, or if there is no question, thank you very much, Dr. Zubair. We are happy to have you with us in this occasion, and I'd like to invite you in near future again.
Thank you.
So please be with us. And now we are at the end of our session. So I'd like to request Dr. Taman Zarin, Assistant Professor of Department of Microbiology, Stanford University, Bangladesh, to have the concluding session. Thank you.
Thank you very much, Professor Dr. Shahidul Kabir, sir. Actually, we have had an extremely useful time from last 2 to 3 hours, and we have learned a great deal and are very much indebted to the speakers who have discussed the host pathogen interaction in inflammation and infectious diseases. And I'm certain that this will enlighten our knowledge and understanding regarding host pathogen interaction. So I think it's an wonderful success. So on the very first, I would like to thank our distinguished guests, our speakers, who have managed their valuable time to disseminate their ideas and works and provide us a better understanding throughout their resourceful and excellent talk. We hope that this is the way we'll have them beside us in the future. So thank you very much. I would like to thank Stanford University, Bangladesh, especially Dr. Farahnas Firoz Madam, Associate Professor and Chairman, Department of Microbiology, Stanford University, Bangladesh, for providing all the facilities and taking the initiative to make the e-symposium happen. Nonetheless, I must thank Professor Dr. Shahidul Kabir sir for his continuous contributions, support, and communication with the speakers time to time. As well, I must thank my dear friend Aftabuddin, Assistant Professor, who monitored the overall arrangement and programming of the session. It would be incomplete if I don't mention the name of our dearest Kamal Kanto, lecturer, who worked hard throughout the program. And above all, I would like to thank all the faculty members in the Department of Microbiology, Stanford University, Bangladesh, for their thoughtful ideas and staying along the session with us. All the audiences and students, thank you for your joyful presence within such a short notice. And we have received many questions, but due to time limit, we couldn't ask all of them. And we all learned something new today, something we didn't know before we joined the session here. For students, I hope this will encourage you and make a challenge in you to learn more about this interesting issue. So today's talk might help at least few of you to choose your field of research in higher education. So choose something that makes you interested or encourage you, and let's think more, as thinking may bring new ideas, and new ideas may bring the positive changes in the society and ultimately in the world. Thank you all to patiently listen to the session, and I wish you all the very best. Thank you again for being with us today. Thank you very much.